Welcome, everybody. I'm Dr. Richard Winegar, Chief Scientist at MRIGlobal. I want to thank the Stowers Institute and MRIGlobal for providing this opportunity to speak to you about the topic of Disease X, COVID-19, and the evolution of testing. You may be thinking to yourself, what does COVID-19 and testing have to do with evolution, and just what is Disease X? Like a lot of people, you may think of evolution as a remote topic that relates to events long ago and far away. However, when it comes to emerging diseases, evolution is a living process affecting our daily lives. The better we understand this, the better we will be at testing diseases and detecting new threats before they evolve into pandemics. In the past 2 decades, there have been numerous public health emergencies and pandemics that had various origins. In 2003, we had the SARS epidemic, which was followed in 2012 with MERS and the current 2020 novel coronavirus pandemic. And each of these involved the novel spread of an animal virus to humans. In 2009, we had the emergence of pandemic H1N1 influenza, which emerged as a new strain of an existing disease. And then with the Ebola outbreaks of 2014 and 2018, we have reemergence of an existing disease. And then a fourth way that a pandemic can arise is through global spread of a regional disease, which was the case of Zika. And if you think about the frequency of all the diseases we've had in the past 2 decades, it's not unreasonable to think that there will be the risk of something new emerging in the future. And one way that this is captured is the concept of Disease X, which means that at any time we can expect a new emerging disease for which we don't have the public health capacity to be as responsive as we would like to be. To give you some background on Disease X, the World Health Organization annually determines which diseases pose the greatest public health risks. Examples include the current COVID-19 pandemic. There's also Ebola and Marburg diseases, Lassa fever, MERS, SARS, and Zika. In 2018, the WHO began adding a new category, which was Disease X, which acknowledges that there are diseases that aren't on our radar. And the hope is that by acknowledging this fact, this will help the global community to prepare for these unknown threats. So how do these new diseases evolve? Well, most pandemics involve the transmission of a virus from an animal host to humans. Sometimes this may be a wild animal, such as bats or rodents, or it could be domestic animals, such as chickens and pigs. However, most viruses are well adapted to their animal hosts, but sometimes a rare event may trigger a human pandemic. For example, the 2009 pandemic influenza began through the simultaneous infection of a swine, avian, and human flu viruses. Parts of these viruses combined to produce a new strain, and the human population as a whole lacked immunity to this new strain. So greater ability to cause widespread disease than a normal seasonal influenza. A second example is the novel coronavirus. And for all of these viruses, the bat is a natural reservoir. The virus can infect an intermediate host, which might be a domestic or game animal. Humans become infected after contact with the intermediate host, and this might occur in a place such as a food market. After the virus infects either the intermediate host or human, the virus can evolve under the selective pressure of that host, and it may gain properties that increase its virulence or its ability to spread. I think we can safely say that COVID-19 meets the criteria for Disease X. What we've had is a sudden emergence of a disease for which the global public health resources were not able to respond to. As of a few days ago, the total number of global cases was over 39 million, with over 1 million deaths. And then within the US, we've had over 8 million cases with over 200,000 deaths. And the sudden emergence of a novel global pandemic has stressed the entire health infrastructure. So there's been intensive efforts to develop the tools necessary to fight the pandemic. This includes various treatments, a number of vaccines, as well as developing the means to do testing for various purposes. Next, we'll go into a little more details on the COVID-19, its virus, and what infection involves. So in the media, you will hear a lot of different words surrounding the virus, the disease, and testing. And what I'm going to do is walk through a few of these key terms, since they are important for you being able to stay informed when you hear these words used. So the first term is virus, which most people will have encountered in school. But as a quick reminder, a virus is a submicroscopic infectious agent consisting of genetic material that can be either DNA or RNA that is in a protein package. Unlike bacteria, which can live independently, a virus can only multiply in a living cell of a host. So a human virus will attach to a human cell, inject its genetic material, and then the virus hijacks the host cells to make more copies of itself. And then large numbers of the newly made copies of the virus are shed from the infected cell, which can then infect more cells or infect another person. The next thing I wanted to go over is distinguishing the terms relating to the virus and the disease it causes. So the term SARS-CoV-2 virus is the virus that causes the novel coronavirus infection. And you can break down this viral name into its components. So the SARS part stands for severe acute respiratory syndrome. CoV stands for coronavirus, and then 2 indicates that it's the second virus that causes a SARS-like disease. Now, COVID-19 is the actual infectious disease. It stands for coronavirus disease, and 19 stands for 2019, the year that the disease was first identified. And we'll go into a little more detail about the SARS-CoV-2 virus itself. So you're probably very familiar with this image of the virus, which appears as a ball that's surrounded by spikes. And if you look at the underlying structure of the virus, you can see that, yes, indeed, that the virus particle is surrounded by these spike proteins, which are embedded in a membrane protein. And then inside of all this is the RNA, which is the genetic material of the virus. Now, the virus uses the spike proteins to attach to lung cells in humans. By binding to that human cells, it's able to fuse with a cell membrane and inject its RNA. The RNA then directs the infected cells to make more copies of the virus. The virus copies then can exit the cell and infect more cells. Eventually, this may progress to COVID-19 disease. And in response, the body will start making antibodies to the virus to fight the infection. And as you can see, testing can actually take advantage of various components of the virus as well as the body's immune response. So for example, molecular tests will detect the RNA. An antigen test will directly test for the presence of the spike protein. And then an antibody or serology test will detect the antibodies that the human immune system produces in order to fight the infection. In this case, we have an antibody that is specific for the spike protein, which in turn blocks the virus from being able to infect the human cells. Disease stage of COVID-19 affects what kind of testing is appropriate. Initially, testing for the virus itself is most sensitive. So this usually takes the form of nasal swab. It will be an early stage viral in order to be able to be detected. But over time, the virus levels will increase. This will cause molecular tests to have increased sensitivity. But over time, as the body starts to fight infection, antibody levels will rise, causing a corresponding drop in the levels of the virus. So as this occurs, then antibody tests become more sensitive. So then you'll switch from nasal swabs, which are intended to detect the virus directly, to using blood samples, which can detect antibodies produced by the body. You will note that there is a period between the declining level of the virus and increasing levels of antibodies in which both tests may not perform as well as would be preferred. Let's discuss a little more detail about various testing methods. The first category are molecular tests designed to detect viral RNA. RNA is extracted from a swab, and then the RNA is subject to something called polymerase chain reaction, or PCR, which is the most commonly used molecular method to detect viral RNA, although there are other similar assays that produce exponential amplification, but all of them have some features in common. So the components of the PCR include short DNA primers that are matched certain regions of the virus. There's also replicating enzymes, which will then use those primers to produce new copies of the virus RNA. And there's a fluorescent detector. After going through cycles of heating and cooling, we produce exponential amplification. So after 40 cycles, you can go from having one copy of viral RNA to 100 billion copies. And as those copies increase, the amount of light that they produce increases. So that makes it possible to have a threshold such that when a certain amount of light has been produced, we can say that viral RNA has been detected. Now, one thing to consider is that the evolution of the virus may interfere with molecular tests. So a molecular test requires that you have knowledge of the virus genome, because you have to design primers and probes that match the virus. So for example, here we have one of the two primers for COVID-19 test. What can happen is that viruses may mutate over time, causing mismatches with these primer or detection sequences. Some of these mutations are called deleterious mutations, and they are essentially lethal to the virus and thus will not replicate and spread. However, there can be random, what are called neutral mutations that don't affect the virus. This figure highlights in green real-world examples of circulating viruses that have to 19 molecular tests. And then the last category are adaptive mutations, which can represent a shift in the properties of the virus. So the virus may no longer resist drugs, or it may be no longer recognized by human antibodies. Then any of these mismatches with the virus genome can cause a molecular test to perform poorly or cause failure. The second category of tests are antibody, also known as serology tests. A serology test detects the presence of antibodies produced in response to COVID-19 infection. Serology tests are very important for several reasons. First of all, it enables diagnosis of later stages of infection. It can also be used to determine the percent of the population that has immunity, which is sometimes known as herd immunity. And it also is a means of determining the efficacy of a vaccine. So you can determine whether being vaccinated actually produces the type of antibodies needed to fight the virus. Evolution plays a role also in this aspect of the viruses versus the immune system. So the immune system contains antibodies present as a large pool of proteins with random binding sites. So here we have just 3 different cells, each producing a different antibody, of which only one of these is binding to the virus of interest. And since this cell has bound to the virus, it is stimulated to produce more copies of itself. And then these copies then can produce even further copies. And each of these immune cells will produce antibodies. So over time, the body will make more cells, and those cells will make more antibodies. So it's very much like adaptation that we normally associate with evolution, but this is inside the human immune system. And later on, if the virus is encountered in the future, there will be memory cells which are geared toward mounting immune response if that virus is ever encountered again in the future. Now, the virus itself also has ways to counter the immune system. The virus can undergo random mutations that change the parts of the virus that the antibodies recognize. So there's a chance that by a random mutation, the antibody will no longer be able to bind to the virus. And viruses that can escape the immune system can then better infect cells and produce more copies. So there is a selective advantage for viruses that can escape the human immune system. The third category of tests are antigens, or what are often called rapid tests. So antigen tests directly detect proteins on the surface of the virus, and the specific molecules on the virus that's recognized are referred to as antigens. So for SARS-CoV-2 virus, the spike protein is commonly used as a potential antigen. An antigen test is very similar to a pregnancy test. So you can add a small droplet of sample onto a strip that contains antibodies to specific proteins on the virus. If the virus is encountered in the sample, the antibodies will capture the viral particles, and then that will cause detector antibodies to bind to that same site. So you'll end up with producing a positive result at a specific site. So this is very similar to a pregnancy test and only takes a few minutes. Here you can see antigen test before any sample's been applied. This is a positive result where we have the second band. If the patient is negative for presence of the viral antigen, it'll only produce this control line and not the detection line. So some things to keep in mind is that antigen tests aren't as sensitive or as accurate as molecular tests. So they're most useful for screening potentially infected individuals. So it's likely that anyone who gets a positive result by the antigen test is receiving an accurate result, but negative results may need to be confirmed with a more sensitive molecular test. And as with serology tests, mutations may cause a mismatch between the antigen test and the virus. So now we're going to go into a little detail about the process by which testing becomes available to the public in the United States, which in recent months has been through the FDA's Emergency Use Authorization Program. So as you all may know, the FDA regulates testing that's used for diagnosing infections. And approving a new test can be long and complex. However, in a public health emergency, the FDA allows emergency use authorization, or EUA, of diagnostic tests. COVID-19 was declared a public health emergency on January 31st of 2020. And the EUA is an important tool to provide diagnostics when a novel disease arises for which there are no existing approved tests. The EUA doesn't require the large clinical trials that would normally be required for approval. However, one thing to keep in mind is that an EUA is temporary. Once a public health emergency expires, manufacturers are expected to submit tests for full regulatory review before marketing of the test. It's remarkable the number of tests that have been authorized under the EUA program. As of January 31st, when the health emergency was declared, there were 0 tests available. By February, we had 2 tests, and then the number of tests kept rising and rising. So at one point in June, there were more than 1 test a day being authorized. And as of October 20th, which is 1 day ago when I'm recording this, there have been 248 EUAs issued. One thing that you'll notice from this chart is that at the beginning especially, the focus was on getting molecular tests authorized. So you can see these in blue. And part of this has to do with that molecular tests are the easiest to develop at the very beginning of an epidemic. Over time, we see a rise in the number of serological tests, and this was due in part for the need to better understand what fraction of the population has been infected and the need for being able to do testing for vaccine trials. The lowest number of tests that have been authorized are the antigen tests that are shown in green. So overall, the majority of tests are either molecular or serological. So what is the future of testing? Well, testing can be used for quite a number of purposes. Obviously, there's diagnostics, which is what we've mostly focused on so far. However, there's also uses in epidemiology, such as contact tracing. It can also be used to determine how many people have been infected and what strains of viruses are circulating. Then there is the topic of environmental testing, which includes surface and air testing. So this can be used to answer such questions as, is a plane safe to board? Are social distancing and disinfecting practices adequate? There is now a new concept that actually is being taken quite seriously, and that is sewage testing. The virus is shed through the gut and will find its way into wastewater systems. And prior to being fully treated, remnants of the virus can be detected by molecular tests. And this can be useful when it's unknown whether in a given community, whether the virus may be circulating among those group of people. And then the last area is biosurveillance, which includes testing for the presence of viruses in domestic and wild animals and insects and other vectors of diseases. And we'd also like to know whether viruses that may be found in these animals or vectors poses a risk for infections in humans. And the outcome we would most like is to be able to discover viruses before they lead to the next disease X. Some of the weaknesses of standard molecular tests are that if a novel virus emerges, there can be no assay for detection. So such was the case when COVID-19 emerged, is there were no assays. If the virus mutates, that may cause the assay to fail. The virus may become drug resistant, but a molecular test may give no information as to whether that virus is drug resistant or non-resistant. And lastly, a patient may appear to have one disease, but actually has an infection that's not specifically targeted by the test. Overall, assay design can be complex because it requires 2 primers and 1 detector that must meet the virus, and sometimes the assay may not be as specific as needed. However, there are new technologies that are rising that each partially meets some of the weaknesses of standard molecular tests. The first of these is next generation sequencing, which is high throughput sequencing technology that arose out of the Human Genome Project. And then there is CRISPR diagnostics, which is technology based on advanced genome editing methods. So next generation sequencing, as I said, is a set of technologies that allow high throughput genome sequencing. And this may involve a large scale laboratory sequencers that can do thousands of samples, but it's also possible to do sequencing with little handheld devices that are powered by a USB port on a laptop. Some of the benefits of whole genome sequencing is that it's possible to get genomes of pathogens or multiple pathogens, unlike a standard PCR test, which may only detect one organism. It's theoretically possible to detect any pathogen that may be in a sample. And unlike PCR, you can discover the presence of a novel pathogen. So you don't have to know ahead of time of what that sequence is. You can actually discover a new virus. So that's, in fact, how viruses such as SARS-CoV-2 are initially discovered, is through genome sequencing and finding sequences that don't match any other known virus. So something like that wouldn't be possible using a test like PCR. And it's possible to identify multiple mutations throughout the genome that indicate a new strain or emergence of drug resistance. So unlike PCR or other standard molecular assays, which only target a very small region of the genome, the next generation sequence technology allows us to look at the entire viral genome and understand what all mutations are arising. So NGS provides a global view of both infection and virus evolution. So if you do NGS sequencing of a patient sample, is you get a lot of different information. Through the RNA portion of sequencing, we can get information about the viral genome. We can also get an understanding of what's called the host transcriptome, which is basically providing information on how the body is responding to the infection. And then the DNA portion of the NGS can give information about the patient and any particular risk. So there may be mutations in certain genes of that patient, which may make them more resistant or more susceptible to severe consequences of COVID-19 infection. So NGS provides a lot more information than can be done with most other tests. Another feature of NGS is that when tied to cloud computing, it allows us to perform high volume sequence data analysis and to share that information with researchers around the world. And this has led to a better understanding of the global spread of the SARS-CoV-2 virus and the emergence of new strains. So this slide shows the evolution of viral strains over time since December 2019. And as you watch this video loop proceed, is at the very beginning of the outbreak, there were very few limited strains of the virus, but over time, new mutations arose, leading to new strains, and then those strains became much more predominant. So you can see initially there were a lot of these blue strains, but over time they are overtaken by the orange, yellow, and green strains. And this is only possible because of the ability to do high throughput genome sequencing of the virus. So pretty much every day, thousands of virus genomes are being sequenced and uploaded to the cloud, and allowing us to get an early warning whether a strain may have new properties that might be affecting molecular tests, vaccines, or drugs. Another technology I wanted to talk about is CRISPR. So CRISPR is a technology that probably a lot of you have at least heard of in relationship to its use for genome editing, but it is being adapted for use in rapid testing. CRISPR gained some notoriety this year because of the recent 2020 Nobel Prize in Chemistry for the discoverers of CRISPR, which are Jennifer Doudna and Emmanuel Charpentier. And with this technology, a Cas enzyme, which is an enzyme that's used by CRISPR, is combined with a special guide RNA sequence, which allows a specific sequence to be found and cut. And you can take advantage of that cutting to produce color or light. Assays can be reprogrammed with new guide RNAs on demand if new strains or new viruses arise. So unlike PCR, which requires 2 primers and a detector, a new CRISPR assay only requires the design of a new guide RNA. And it can be performed on a paper strip, similar to a pregnancy test, or it can be automated to perform thousands of different tests. DARPA, Defense Advanced Research Program Agency, has funded an initiative known as DIGIT, which is meant to really progress the technology of CRISPR for producing diagnostics that can be used in a wide variety of situations. Another area I wanted to talk about is point-of-need home testing. So right now, public health measures such as social distancing and isolation make large-scale frequent testing difficult. And part of this is that the majority of authorized tests are what are known as high or moderate complexity tests. So for these tests, a person must go to the doctor or clinic and give a sample. The sample then may be shipped to a centralized or hospital lab. and it may take several days before you get results. And there's limited availability to non-symptomatic individuals or the general population. So as I was saying, nearly all tests that have been authorized are either high complexity as shown in red or moderate complexity as shown in yellow. So only 14 tests have been authorized that are known as waived or simple complexity tests. So what would the advantages of a point of need or home test be? So first thing is that this is a simple test that can be performed at home or at a place of work. The tests are inexpensive enough for frequent screening, and the results can be obtained in 15 to 30 minutes. And sometimes you can use non-invasive samples such as saliva. For point of need tests, these are low complexity tests that are suitable for use in a doctor's office, clinic, or airport, and they can get results in less than an hour. Point of need tests fall into several categories. There are rapid antigen tests, which for the most part are very similar to pregnancy tests, but they may use a smartphone as a reader or a small handheld device to provide results. And then there are a few molecular point of need tests. These take a little bit longer, about an hour, mainly because the kit instrument performs RNA extraction and then performs the molecular amplification and reporting of the results. But overall, compared to the very high complexity tests, these give results in very short turnaround time and expand the number of places that the tests can be performed. And then finally, there is only one low complexity test for serology that's been authorized, and it produces a result in 10 minutes. So unfortunately, unlike tests that are authorized for places of work or airports and other situations like that, there are currently no authorized home testing kits. What we have instead are kits that are available for home sample collection. So for these tests, the patient uses the kit at home and mails in a swab to a lab to perform the molecular testing. So the advantage of this is that it is compatible with social distancing, so you don't have to drive to a clinic or your doctor's office to provide your sample. You can do that at home. However, on the downside, it may take several days to get the results. And these types of tests are mainly used for employees in high-risk occupations such as airlines or grocery stores. In conclusion, I hope you come away with a better understanding of testing and that testing have different intended usages and different advantages and disadvantages. So we have the molecular tests which detect viral RNA and can be used during early or mid stages of infection. Serology or antibody tests which detect human antibodies and are good for mid to late or even after recovery. And then antigen tests which directly detect viral protein and are mainly useful during the middle of infection when viral levels are at their highest. Testing will need to evolve to respond to current pandemic and future disease X threats. But I do want to assure you that MRIGlobal is actively engaged in driving the future of infectious disease testing. In our Diagnostics Center of Excellence, we provide services ranging from rapidly evaluating new tests for EUAs to developing and validating new diagnostic technologies. And we are also developing the infrastructure to allow disease surveillance globally by providing and staffing mobile labs and even designing labs that fit in a suitcase. Well, I want to thank you for participating in this presentation. I hope that you will watch the complete set of videos prepared for this series. My colleagues have much more information regarding the full range of activities that MRIGlobal is engaged in. If you have any questions or comments, please feel free to use the contact information on the slide. And thanks again, everybody.